I'd like to introduce Jake Gendron to the group. Jake hails from Biddeford, Maine, and he received his BS degree in biology with a minor in chemistry from the University of Maine, and somehow matriculated all the way to Texas Tech for his master's, where he worked with Dr. De Los Reyes in elucidating genetic networks within inbred rice for drought stress conditions. He joined my cotton breeding program as a Salyer Fellow in 2018, and he's working in the area of fiber quality. Dr. Eric E. Kay with the Fiber and Biopolymer Research Institute in Lubbock at Texas Tech is a co-chair of his committee. And today he's going to give us a bit of an update of the fiber quality components that he's working on. With that, Jake, it's all yours.
All right. Thank you, Dr. Smith. Can you guys hear me?
Can hear you well.
Yes.
Okay. Thank you. Okay. Y'all can see the screen?
Absolutely.
All right. Thank you again, Dr. Smith. With that, I'll be discussing the 3 projects that I'm currently working on in the Cotton Improvement Lab. So just a little bit of background before we get started, like always with these things.
Jake, you may want to make your slide full size.
Are they not?
I see 2. I see a.
Does this work?
Yeah, that's right.
Okay, sorry, I just had the presenter view up. So the United States is one of the top global producers of cotton, only behind China and India. We produce about 20 million bales on average a year and have for the last several years. Now in the United States, we grow 2 major species, Gossypium hirsutum, which is the upland cotton, and Gossypium barbadense, which is the Pima. The majority of cotton grown here is the upland cotton variety, but Pima also has some intrinsic value due to its higher length and strength potential as opposed to the upland varieties. In Texas alone, we grow about 30 to 40% of all the cotton in the United States. And of all the cotton in the United States, roughly 2/3 of that is exported. So the US cotton-based market is largely export driven. And that's relatively important because over the last decade or so, China, the #1 importer of US-based cotton, has drastically cut its imports. There's been a global increase overall for these petroleum-based clothing, the synthetic fibers that you have, the polyesters. And within China itself, they've focused particularly on improving their own agricultural programs, as well as introducing a cotton stockpiling program. And despite these cuts to imports, China still accounted for almost 40% of the world trade in terms of textiles and apparels in 2013. So given this, I think there's a major problem that cotton breeders need to address, and that's how do we keep US cotton competitive on a global scale given these trends? So breeders have a number of different tools at their disposal to improve cotton fiber and then downstream effects of overall improving yarn quality as well. Those 2 tools that we use most readily are what's known as high volume instrumentation, or APHIS, shown here on the left. High volume instrumentation is the predominant means of data collection right now. It's used by both breeders and gins to assess bundles of cotton samples for a number of different factors, including grade, leaf, but other outputs that are important to breeders include bundles strength, length, uniformity, elongation, and micronair. And it does so at a very high rate of speed, which is why it's so popular among so many people. In the last decade or so, there's been a more recent focus on APHIS measurements. APHIS, however, takes a little bit longer time, but it also gives us a broader range of measurements to look at. Instead of measuring particular bundles, it measures the individual fibers themselves. And it's been shown that some of these individual fiber measurements have given us a greater predictability of yarn quality overall. Now, on top of these sort of tools that breeders have always used selection indices too, and it's not different in cotton. We've aggregated HVI or APHIS parameters to try to develop some sort of scoring mechanism to ease breeders' selection for improving fiber quality. In particular, one of the more well-known ones is known as a quality score, which was developed by Dr. Fred Borland out of the University of Arkansas. And he developed this a while back, I think it was 2010 or so, and it was designed to improve cotton fiber selections regarding fiber quality and loan value. And in particular, people have been curious about whether or not some of these quality scores can be used to predict yarn parameters further along, but the results have been generally pretty mixed. And so it's also convoluted by the number of different spinning techniques used and environmental effects that we see out in the field. It's important for breeders to kind of bridge this gap between the selections that they're making for improved fiber quality and then the downstream effects of having improved yarn quality as well. So there's a lot of data on this slide, and I just want you guys to focus in particular on the correlation matrix highlighted in the green boxes here. Dr. Smith and I were particularly curious in this concept of explaining some of the yarn parameters through HBI and APHIS. And so we went back through an old graduate student's data set. And in particular, what we looked at were these 129 varieties, which had been released over the better half of this last century. And in particular, we were looking at correlating some of those release years, in particular with the HBI parameters, as well as the quality score measurement I kind of mentioned briefly before. And as you can see here, the correlations between those HBI parameters and the release year, the varietal release year, is pretty strong. They're significant at 0.001 for all these correlations. And so as breeders overall, we've been making selections for improved fiber quality. But when we look at how this relationship translates into improvements in yarn quality, we see that these necessarily haven't improved our yarn quality overall over this time period. So given this, we have this disconnect of trying to determine yarn quality parameters from HBI or APHIS measurements. And so we need to kind of address this as breeders going forward to find a better way of bridging this gap. One means in particular, and this project was brought to my attention from Dr. Kay up in Texas Tech, we decided to look at what's called propensity to break. Now, propensity to break is a relatively new concept, and I'm going to keep explaining as we go along, but essentially varieties were identified by Dr. Kay, 12 different varieties in a population of individuals, and he classified them as either having a high or low propensity to break. And essentially these individuals were thought to be more or less susceptible to the mechanical breakage that's often incurred by fibers during the ginning to spinning processes. And so in particular, we were interested in whether or not we could mimic some of the mechanical fiber damage that's experienced by these cotton fibers, and whether or not we could accurately capture that data and then use it to make future selections. So what is propensity to break? It's a relatively new measurement, like I said, and we don't really have a standardized way to evaluate it. Like I mentioned before, we have APHIS numbers that are given to us from samples. And one of the APHIS data points is what's known as a fiber length distribution, shown here in these bottom two figures. And what it basically tells us is the distribution of fibers by length for a given sample. So this 6% here that you see corresponds to 6% of the sample is at a length of 0.0625 inches. What we do is we take these distributions, or we take these samples, sorry, and we run them through what's known as an MDTA3 cleaning process. And this MDTA3 cleaning process was designed to extract trash from cotton samples. So it's a rather aggressive process that has been shown to damage fibers and cause significant increases in what's known as short fiber content. And what short fiber content are those intervals of cotton fiber that are less than ½ an inch in length. And so we run this fiber through this MDTA3 cleaning process, and we get these length distributions before and after. And what we do is we calculate the distance between these two distributions, and then we perform some type of either hierarchical clustering or k-means clustering. And what we should expect to see is that these groups should be separate from one another, that we should have high propensity to break grouping differently than low propensity to break. And just for an example, this distribution that you see here is what I would consider a high propensity to break. You have an accumulation of shorter fiber content. As you can see, that 6% that I was saying you earlier is now 8.5% roughly, and the distribution of these fibers skews left. So this would be something that would be bad that we're not necessarily looking for in the data set. So I looked at the 2018 data that we had, and I calculated those distances across a number of different methods. And essentially, I was just curious in how these methods might affect how our clustering was happening. I did k-means and single as well, but didn't show them because they were either redundant, or in the case of single, it was slightly messy. But as you can see here in our 2018 data, there's no distinct clustering between these high or low propensity to break lines. It tends to be generally pretty mixed across the board, as you can see, no matter what method that we use to calculate these distances. And there were some environmental effects that we could kind of explain some of this away in the data set. So moving on to the next year, unfortunately in 2019, the data was processed incorrectly. And by the time we realized it, we were in the middle of COVID and it was a little too late to go back and fix, unfortunately. But we did get our 2020 data back here recently, and I was able to calculate the distances the same way that we had before using the same methodologies. But again, you see this same sort of problem arising, that we're not really seeing this distinct clustering of high or low propensity to break varieties separate from one another. There tends to be a mixture of all of these varieties together. And so when you get these sort of results, you start to question what might be happening, what's going on that could sort of lead to this thing. And in particular, as many of you may recall with 2018, we had one of the wettest harvests on record. And in particular, that definitely affects cotton quality overall in the field. When the bolls open and they're exposed to about a month and 30 inches of rain, it's just going to absolutely decimate your cotton fiber. But that doesn't necessarily account for why we have this data set in 2020 where we can't really explain these differences between our high and low propensity to break varieties. So I asked myself in particular, is there something that we're missing here? Could we group the data differently? And were the correct varieties sort of initially identified? In particular, I went back through and I want to reemphasize the point here of what we're looking at. So up here in the first graph above, it's bad or high propensity to break. And as you can see, or as I showed before, you have this distribution of fibers that skews left after this MDTA3 cleaning process. And in particular, what we're looking for the low propensity or these good varieties is to have a distribution that looks almost exactly the same after as it did before this MDTA3 cleaning process. And so in particular, what I started to realize is that instead of calculating the distances between the entirety of these two figures, that maybe we should have a particular focus paid attention only to the short fiber content, those fibers, like I mentioned before, that are less than ½ inch in length. And the reason why this is important is that those fibers that are less than ½ inch in length generally aren't spun into yarn. There's, I think, one technology that kind of utilizes it, but it's not widely used. The two main methods are ring and rotor spinning, and neither of those, as from what I know, can utilize those fibers that are less than ½ inch in length. So knowing this, instead of calculating just the distances across the entire lines, what I decided to do was look at the sum of those distances, but just for those fibers that were ½ inch in length or less. So in particular, we were looking for, or what we were expecting to see is that those fibers that should be a low propensity to break should not be accumulating any more short fiber content. And that would indicate a value closer to 0 here in these tables below. And then those varieties which have a higher propensity to break that are accumulating a much higher degree of this short fiber content as the distribution skews left, we should see a larger negative number. And as we look through the data set here, what you can kind of see is that there's not a whole lot of differences initially between those lines that were identified as high and low propensity to break. I calculated just the averages across years to give that effect that the differences are relatively minute. There are some lines that you can point to here and there that seem like they would fit either a high or low propensity to break. But overall, this might explain why we're not seeing these distinct clustering patterns in our hierarchical clustering system. So, knowing this, I decided to just go forward and just put some statistics to what we were looking at here. And as you can see, the 2018 data set was the outlier of a year. And obviously we can explain some of that data set with the environmental factors that were occurring at the time. I also looked at by variety, the changes in the sum of the short fiber content. And what I noticed was that there is no difference, whether it be across 2 or 3 years, whether you remove that 2018 data set or not. And I know it's a generally small sample set to do this forward analysis with, but it just to drive home that point of what we're looking at and the fact that there's no differences here. So that being said, I think going forward, we need to reassess how the varieties we used in this study were characterized and how we measure and define a high and low propensity to break. I think secondly, we need to maybe place a little bit more emphasis on the change in short fiber content rather than looking at the entirety of the line distribution and calculating those distances. A particular emphasis on short fiber content is essential, I think. Further emphasis will also be placed on maturity with regards to how do we analyze the data set. Maturity has a significant effect on obviously fiber development, and the HBI and APHIS data sets give us, or sorry, APHIS data set gives us a maturity ratio as well as a maturity distribution, which we can look at and sort of analyze alongside these factors. So going forward, that's one other thing we're going to look at, but I don't think it's going to explain away a lot of the variance that we're seeing here in the data set. And lastly, I think given the apparent importance of short fiber contents in determining propensity to break, and their importance in terms of some literature has cited their usefulness in predicting yarn quality parameters. I think that short fiber content should be used as a selection tool going forward and in any future cotton fiber indices that generally get developed. So with that said, I'll move on to the second project. And this is just going to be sort of an overview of these two projects as we're a little bit short on time here. The second project was to examine within bowl yield components for a set of 267 progeny rows, and to elucidate this relationship between elite varieties with high fiber quality, but low lint percents and poor elongation. And so we have a replicated CRD and HBI measurements over 2 years. And this is handy to us because when we're looking at the data set, and the reason why this is important is because when we're making these sort of improvements in fiber quality, and I showed you guys this figure before, this correlation matrix earlier, but when we're improving our length, uniformity, and strength, as you see here, this often has a negative corollary effect with some of the elongation factors. And elongation in particular has had a recent, I guess, revitalization of interest because it's been seen that elongation might have some important correlation to some of the yarn parameters that we're also looking at. And in particular, lint percent as well is important to a lot of breeders because lint percent is directly correlated or is an aggregate for how we calculate yield in the field, I believe. And so being able to select for varieties that have this improved strength and length potential while mitigating the losses for elongation and lint percent are going to be essential to not only improving fiber quality, but yarn quality as well. And so there's some literature out there that sort of suggests that some of these negative correlations, these low correlations, might be partially attributed to the repulsion phase and linkage. And so it's our theory that we might be able to simultaneously improve these varieties for both of these or all four of these traits. It's a little bit of a process to get all the data for these within bowl characteristics that we're trying to measure. And so I've just sort of listed the steps here of what we're trying to accomplish right now. We've obviously collected our samples from the field and I've gotten weights and counted the number of seeds. And right now I'm at step number 4, which is calculating the seed weights before and after we delint. And then we'll finally move on to calculating the seed surface area based off of volumetric displacement. And what that will do is give us the right values to calculate these parameters here on the right, as you can see. And then once we're able to calculate these parameters on the right, we'll be able to correlate it back to those two, and then finally this third year of data that we're going to be getting from the field. And it should be interesting to see whether or not there's varieties or we can expand or elucidate any more of this relationship between high strength and length potential in lines while mitigating this elongation and poor lint percents factors. With that said, I'll move quickly on to the final project that we're currently working on right now. It's a line by tester, and we crossed Altec 7A21 as our standard quality tester with 12 TM strains. And these strains had generally a high upper half mean length and strength relative to the general breeding population. And our goal was to determine combining ability, heritability, and to conduct a GMA to examine crosses made for improved fiber quality, in particular strength and length. Now, this being said, in 2019, crosses were made between Altec 7A21 and the 12 TAM varieties with good length and strength potential. This last year, we planted our F1 self and back cross with both parental lines and reestablished the crosses we had made in 2019. We got our HVI measurements back recently, and while I've started to analyze the data sets, we don't have a complete picture yet, and I'm still in the process of calculating heritability. So going forward, that's something I'm just trying to work towards completing. But in 2021, we'll be replanting that family in a replicated RCBD in College Station. And from there, we'll be able to calculate a generation means analysis and reestablish those estimates of heritability. So with that, those are the 3 projects I'm currently working on in the cotton lab. The goal is to hopefully graduate this fall. And I'd like to give a special thanks to Lori and Jana Love at the USDA. You guys have been very helpful to me during this whole COVID process and allowing me to use your facilities. I promise you I will return that roller gin if Dr. Haig doesn't burn it to the ground first. I'd also like to thank my other committee members, Dr. Smith, Dr. Haig, Dr. Hickey, and Dr. Stelly, as well as the graduate students in our lab, Allie, Braden, Caitlin, and Josh, our technicians and our undergraduate workers, Texas A&M AgriLife, and the FBRI up at Texas Tech. And with that, are there any questions?
Good job, Jake. Number 2, questions?
Take number 2. Scare them into silence. All right. Oh, is there some in the chat?
Yeah. Alexandra has one.
I never threaten the roller gin, says Dr. Haig. Just my life. Okay. No. So Ali says, how does short fiber content correlate with upper half mean length? And do you think that upper half mean length in combination with uniformity might give us a better view of your propensity to break? So upper half mean length is a measurement of HVI. And in particular, what APHIS does is APHIS measures those individual fibers and gives us what's that length by number. And so that length by number is how we calculate the majority of other length parameters for the APHIS calculations, if I recall correctly. And when we process samples through HVI, if I am recalling this correctly, I think a lot of those shorter fibers get missed because you're analyzing the bundle as a whole, as opposed to those individual fiber measurements. So how does it correlate? I don't know. That'd be a great question, and it's something that I can kind of run relatively quickly here to kind of look at. And do you think that the upper half mean length in combination with uniformity might give you a better view of your propensity to break? Yes and no. I think right now, I think the focus will be on maturity. Actually, I can just go to it if it works here. These are the varieties that we're currently looking at right now. And what I decided to do, just out of caution in case this kind of question came up, was to plot the histogram distributions of all 3 projects that I'm currently working on relative to what the loan discounts are and premiums at the gins. And we're already kind of working with varieties that have this high length potential and high overall uniformity. So maybe not, but it's something I could look at in the future. Does it make a lot of, okay.
I got a question while you're reading, and I'm curious if we reduce the propensity to break by 70%, if Dr. Haig and Dr. Smith combined and produced a new line that had a 70% less propensity to break, how would it be marketed and what advantage would it carry in the market?
Well, I think as you can kind of see with this, there's not, so how we would market it would be that we would have to start marketing towards improved yarn quality. There's a general focus right now in cotton breeding specifically on just fiber quality alone, with less of an emphasis. And this is in my opinion. There might be other cotton breeders who differ from what I'm saying here, but there's a heavy focus on these cotton fiber parameters, in particular between the farmers and the gin, because the farmers generally only care about what the gins want, and the gins only want certain parameters that don't necessarily translate to improved yarn quality. So I think it's more about bridging that disconnect and emphasizing that downstream, yes, we might be creating these better quality fibers, but the end goal is to produce higher quality yarns overall, because those higher quality yarns are going to be what's really asked for by a lot of these mills. And so that would have to be the first change in focus. And And how you would get them to do that, I don't know. In my short experience of working with farmers, getting farmers to change their practices and methodologies is probably one of the most complicated tasks that breeders have. So I'm not entirely sure going forward. And what was the second part to your question?
Well, just how you're going to get it to market. Because it's my observation that if you pay farmers $15, they'll get there pretty quick.
Yeah.
And so is there any advantage to them for producing this higher quality thing?
On
Or
a
is
global,
all the advantage to the mill and the yarn manufacturer?
I think on a global scale, it is important, but just on the local scale right now for what we have, it's the relationships between the gin and the farmers themselves, and the relationship between the gin and the spinning is an entirely different relationship. So how we would bridge that would be rather complicated without getting the mills to either change their practice of how we maybe give premiums and discounts. But it's also, we'd have to find a better way to bridge this gap between, these gins use these HBI parameters to assess a lot of the fiber coming in and to give farmers either these premiums or discounts. So you'd have to kind of change the whole system, unfortunately. And that requires us to find a better correlation between these cotton quality scores and our improved yarn quality. Can you explain why fiber strength and propensity to break are different measurements? It seems like they're very similar traits. Good question. Oh, sorry, Leah, I missed your question. I'll get to that here in a second. So fiber strength in particular. So how to phrase this question? The fiber strengths in particular are just measuring the overall strength of the fibers. And when you're measuring the propensity to break, you're kind of getting a multitude of different factors. You're not only getting strength, but you're also getting length as well. You're also getting a little bit of elongation, because the more elastic some of those fibers are, the less likely they are to break. So when we're measuring these propensity to break values, it's a lot more of an accumulation of various traits, if that makes sense to you. I may have missed this.
Jake, could I interject that
Yes.
fiber strength is fiber bundle strength, and so it's the strength of a bundle of fibers, whereas breakage it would reference individual fibers. So they could be highly likely to break as an individual fiber, but still give you a strong bundle strength.
Okay, yeah, that's a good point. Sorry, I may have missed it. But there are certain environmental conditions that also contribute to fiber length and propensity to break. You mentioned this. Generally, from what I've seen, rainfall is one of the most important factors that affect cotton yield. The minute bowls open, there's a number of factors. There's rainfall, there's UV, and then obviously how long those bowls are exposed. You have a greater amount of trash content collecting from the fibers. So there's a number of different ways, but I would say the predominant one is rainfall or water, and then secondary, maybe UV. But other than that, just the general over time, the fibers will continue to degrade. Is there any other questions? I don't see any.
Yeah, there's one by Sam. What are the major drivers for this kind of research?
Oh, sorry, Sam. You mentioned China's corner on the market. Do they have varieties to outperform ours? That's a great question. It's hard to know, but I don't believe so. I think that a lot of there's not always a lot of information that comes out of China too, with regards to some of the varieties that they develop right away. But I think the export market seems to be what's driving this right now. And in particular, obviously the increase in synthetic fibers, your polyester shirts and whatnot, have been kind of changing the market here in the last decade or so. And I know that in particular, Cotton Incorporated is starting this new push against microplastics in the environment that are caused by these polyester synthetic fibers. And they're trying to get companies to push more towards going back towards cotton. Other than that, I can't think of any right off the top of my head. and of other major drivers. So unless Dr. Smith or Dr. Haig, you have something to add that might. No, yes, no. I don't think so.
No,
Nope.
I would only add that relative to China and their capture of the market and how much they produce, economics is more important than the quality of the fibers that they produce or we produce. How cheap can they make yarn, turn it into textiles, and sell it to you?
Other questions? Going once? Going twice? Going 3 times? Thanks, Jake.